Good morning, ladies and gentlemen. Let's start. Let's wait a couple of minutes to make sure everybody is in the room. Welcome to Biology 1500, the Investigative Biolabs. My name is Mark Sharvari. I'm the director of the course and also the lecturer, along with Dr. Dean Coe in the back. And we don't try to teach you everything in this course. You are taking so many other courses that you are learning a lot about biology. We don't want to repeat everything. That's not the point of this course. Those are not the learning objectives. The learning objectives are to teach you laboratory skills through different modules. And we have 3 major modules. In the first 3 weeks, we try to fill your scientific toolbox so you have all that information, all that knowledge, all those skills to be able to run experiments. And that your experiments, those experiments when you are the scientists, are going to be in antibiotic resistance, working with algae, which we can relate to biodiesel production through climate change, and also working with your own microsatellite DNA to learn more about yourself and your ethnic background. These are current topics in biology. Again, we don't try to teach you everything about them, but teach you enough that you can explore more in this course. So these 3 modules are going to cover most of the semester. However, in the first 3 weeks, you are really focusing on those skills that you need to learn in order to be successful during those modules. So again, make sure you attend the labs, and then you become familiar with all those skills. In order to make sure that we teach you properly, at the end of that 3rd week, we are going to have our first practical exam. We don't have prelims in this course, yay, but we have practical exams, 2 of them. So since it's a laboratory course, we want to make sure that you can walk into a lab and you can do hands-on things that we taught you during the semester. All right. So a few course objectives. It's very important to understand that what our course objectives are. Again, this is a relatively young course. It's still like a child, 4 or 5 years old. It's still teaching it to behave properly. But previously, we had a big survey course. We taught the students everything. We had 700 students in the lecture room, and we taught everything from cell bio through evolution. We don't do that anymore. You take those little pieces from other classes. Here, really focus on those course objectives that we were assigned to by the curriculum committee, and we want to make sure that when you walk out of this class, you have those skills. Because these are skills that you can apply right away. And you will hear it later today, that you can walk into someone's lab mid-semester and say that, hey, I have these skills that I just learned in Biology 1500. Do you have a position? Can I start a research project? This is what I know. My studies in your lab. So these are the skills that you don't need to wait until your senior year to start to apply them. You can apply them instantly. So we try to expose you to realistic scientific questions. As I mentioned, antibiotic resistance, climate change, you read about them in the news all the time. So this is something that's not far away from you or you cannot relate to. We want to teach something that you can relate to as a scientist. And we try to encourage critical thinking. How many of you are pre-med? Or you think you are pre-med right now? Yes? Okay, fantastic. No, I trust you. I want you to be my doctor when I'm 80. If I make it that far. That's why this education is very important. We want to train good doctors. I want to trust you. So critical thinking, if you're taking MCAT, MCAT 2015, everybody scares you with that. That's a big change. So critical thinking skills are important. So we try to make sure that you cannot really teach critical thinking, but we can push you to the right direction. Then, as I mentioned already, scientific toolbox that you can carry with you and use it in other situations in biology. Science communication is extremely important. We want you to know how to find primary and secondary literature, how to find a review paper in the library. Because you need to build your research on that information. So that's one of our goals, that you walk out of here and you understand that. And not only collect that information, but also communicate that information, not just to the public, but also to other scientists. Then what you will start actually today, if you're on Tuesday afternoon lab, is thinking through that scientific process. Something that the laboratory instructors and myself have been trained throughout our PhD education or our graduate schools. That's something that hammered into our heads. You need to start it early enough that you get into that habit. It becomes your second nature to think through scientific processes. And trust me, you do this every day in life. You may just not recognize it. So we help you recognize what these processes are and how you can turn it into your benefit in the laboratory. And then, of course, we have a Discovery Science module right at the very end, the DNA lab, and you will learn how to explore patterns in nature. And very lastly, just give you the opportunity to learn about fundamental biological information. I see usually two responses from students when they are especially incoming freshmen. One, they are afraid to take this course because they are afraid they don't have enough information to run these experiments. Trust me, we teach you everything that you need for being a successful scientist in this course. The other, when students are looking at this course, is like, oh man, I'm not going to learn any biology here. They are just playing around with bacteria. No, you are going to learn enough biology here. So if you're really interested in biology, this is something you will take a lot away from it, and you will take away new information, not just how to turn a knob on a microscope or how to use a micropipetter. We try really hard to make sure that we connect everything to a bigger picture and make you understand larger biological phenomena. So you look at these projects and you're like, ah, man, I've done this before. You know, I played with bacteria in high school. I know about antibiotic resistance. I deal with that every day in my research lab where I work. So I just wanted to demonstrate. I've seen this from Ellen Alda, who's running a science communication center. And it's a great example to compare what you may have done in high school and what you are going to do in this lab. So I need a volunteer who is not afraid of water. Yes, who was the first one? The gentleman back there with the glasses. You guys are volunteering. Trust me, if I ask the same question in like 5 weeks, no hands go up. It's like, oh, not again. I don't want to go up there. So hi, what's your name?
I'm Andre.
Andre.
Nice to meet you.
Pleasure. So here's Andre. Please give him a hand. Our first volunteer in Biology 1500. All right, let's step on the stage. So 2 empty glasses, right? Are they empty?
Yeah.
It's like magic, right? They're empty. So simple scientific project. I'm going to stand over there, and you walk these 2 empty glasses to me, please. All right, you can run the experiment. The experiment is to run the glasses here. Excellent. Thank you very much. Was it difficult?
No.
Anything that challenged you with this?
No.
No? Okay, skills you use for walking that you know how to do since you are, what, -2? Yeah, okay. Yeah. Fantastic. So this was something that you do in high school, right? A simple project, you face that challenge. Can you hold this, please? Don't spill much. All right, a little tiny challenge here. So the scientific question is the same thing, right? Oops, see there? The problem is the same thing. I would like him to bring the glasses to me. And if he spills any, he's failing the course. See why I'm not going to have any more volunteers up here? No, wait, we still have 25 minutes of the class. That's good. All right, fantastic. Give him a hand again. Thank you very much. So before you leave, was this more challenging?
A little bit, yeah.
What did you do differently?
I had to balance them and not make the waterfall.
And there are lots of waterfalls in Ithaca, we know that. Yes. So anything else? Like what, did you do anything differently? The first one, how would you identify?
I had to walk slower to make sure that I actually didn't drop the water.
Okay, fantastic. So you walked slower, and you were definitely more careful with that, right? Great. Thank you very much, Andre. I'm aware that this is a silly little exercise, but I will frequently refer to this because this is something I would like you to do during these lab exercises that we have, during your experiments. Don't think about that, oh, because I worked with bacteria before, it's going to be that easy. Challenge yourself. Make it more challenging. Make it a more challenging experiment. Focus on that, so when you are presenting your poster during the last week, your poster is the best one. Your research project was the coolest one. This is something, a poster that you want to keep and put it in your dorm room because it rocks so hard. So focus on that. Challenge yourself. Don't come back to me and say that, hey, you know, I worked with Chlamydomonas algae before. Why are we doing this again? This is boring. No. Challenge yourself. This is a level when you can make it a more challenging experiment. Don't walk with an empty glass. Walk with a full glass. And be careful what you are doing with that. All right. So now you know what this course is about. And I can talk here forever telling you how awesome this course is. It's my job. So I want you to hear from other people who have actually taken this course and from Colin Kearns from OUB who can tell you how you can apply some of these skills you learned in this course.
Thank you, Mark. Good morning, everyone. While Mark's bringing up my PowerPoint, I'll just say, you can tell you're going to have some fun in this class, right? Yeah. So come to lecture. And I'm really here to back up what Dr. Sharvey is saying, that the skills you're going to learn in this class are skills you're going to use in the lab if you decide to pursue undergraduate research for the rest of your career. So it's no joke. The toolbox you're going to get is you're going to use over and over. And I have some undergrad scientists with me who are going to share about their experience. And I think you'll hear from them also that the skills you get in this class you will carry with you for the rest of your life, regardless, actually, of what career path you take. And why am I saying that? Well, doing research and getting into the laboratory is going to give you a whole host of skills. Some are going to be those analytical tools like using a micropipetter, but some are going to be more intangible things like this list. Communication. You're going to learn really great communication with your lab mates in the laboratory and your TA. You're going to learn analytical skills, obviously, teamwork. And one thing about research, and even in a lab setting or a course setting like this, is you're going to learn a really strong work ethic, because as you know, the old saying goes, try, try, try again. Science usually doesn't work perfectly on the first try, and we repeat a lot of procedures in science, and that creates this work ethic because you have a passion about figuring out a question and generating data and figuring out what those data are telling you. So you get some skills besides the typical ones you might think of in research. I know there are plenty of pre-med students in this room, and Dr. Sharvey already addressed a lot of what's up here. This course will help you be ready for the MCAT when the time comes to take it. So take it seriously. You can learn a lot to prepare you, okay? Our first student that's going to speak is Olya.
All right. Hi, everyone. My name's Olya, and I'm a junior bio major in the College of Agriculture and Life Sciences. And I do my research in the Collins Lab in the Department of Molecular Medicine. So basically, my particular project uses yeast as a model organism, and we try to see the effects of modifications such as phosphorylation on proteins activity and membrane trafficking. So how do vesicles actually get through the cells, and how do they know where they need to go inside those cells? And another thing that I do at Cornell is actually a project team called iGEM. So we do synthetic biology, which I guess is a little more on the other side of research. Instead of piecing puzzles apart, we put things together to make biology tools do something useful for us. So this is through the engineering school, so it's a little bit more of an engineering type of research. And our project this year is modifying bacteria to sequester heavy metals, and we're also building a filter to put those bacteria in. And eventually we compete in the fall, and we actually recruit in December, so if you're interested, let me know. And I definitely want to second what Colleen said about your research lab becoming your second family, because that's definitely true for mine. So.
Next, we have Taylor.
Hi guys, my name's Taylor Heaton. I'm a junior in biology, and I work in the Bonter Lab through the Lab of Ornithology. But my work is mainly done at Shoals Marine Lab, which is our off-campus marine laboratory. It's off the coast of Maine and New Hampshire, and it's a wonderful place if you're interested in marine biology and sustainable engineering as well. So they offer courses and internships, which I encourage you to check out, or go stop by in their office in Kennedy, which is here. So please, check out Shoals Marine Lab. But my research project is kind of two parts. So with Shoals Marine Lab, I'm collecting field data on breeding birds out there. And then with that data, I can take blood samples and also come back and work in the lab during the academic semester. So in terms of research, you're not only limited to doing research during the academic year, you can also explore other opportunities during the summer as well. And so I definitely found my passion for research. I love how it makes me think critically and creatively. And I really hope that you, all of you, or some of you, find that passion too. And on a side note, I also wanted to say that there is a wonderful program that doesn't necessarily have to do with research, but it's more on the service side of biology. It's called Biology Service Leaders, and it's available to any biology major to become a leader and any person to be involved in service events. So it's a collaboration between the Public Service Center and the Office of Undergraduate Biology. And there are opportunities for leadership, coming up with creative ideas to integrate biology and service. And there's also many one-time service events around the Ithaca area. So if you're involved or interested in checking out invasive species and how to solve those sorts of problems, and taking care of our watershed, and also outreach education for kids in science, please check that out too.
And our third student is Robert Goober.
Hello, my name is Robert Goober. I'm a senior, and my research is in the Department of Nutritional Science. I study protein folding, and when protein folding goes wrong, how ER stress, which is what occurs, and how that relates to our metabolism, such as how we utilize fat, break it down, and store it. And also, I just want to talk about quickly CURB, which is the Cornell Undergraduate Research Board. I'm the co-president for it, and basically what we do is everyone here should join our listserv to find out about great research events and how to get involved. And we have huge symposiums to allow students to present their research, and Professor Sarvery actually helps out by teaching students, again, how to make posters. And the event you can see right there is in Duffield Hall last year, where we had 150 students present their work, and about 700 students to 800 attended. So it's a really great thing to be a part of. Also, I'm doing an honors thesis for my work, and research really can be amazing. You can even get a paper published, get your name on a paper, and so it's a great opportunity.
Thank you, Robert. Dr. Sarvery, do we have time for a couple questions, or how are you doing? All right, why don't you guys come up, and then we can take, anybody have a question for the students or for me? Yes?
Just email curb@cornell.edu.
So since Robert already mentioned CURB, I just put this up here. You will see these slides on Blackboard anyway, in case you are interested in joining. I've been working with CURB in the past 3 or 4 years, teaching some of their members how to prepare posters, something that you are going to learn here in this course as well. Do this. There are 3 types of poll questions that you are going to face this semester. The one are in-lecture poll questions. They are going to be questions about your readings both in lab and lecture. Again, I don't want you to think about those 2 things separately. This is if you will, a lab lecture. Okay, so your readings for that week are important, and I may ask questions that are in the lab manual, not just in the live book. They need to be answered in lecture, during lecture time. If I actually see that you answered it from outside, there will be consequences. There are in-lecture survey questions, questions that there is no correct answer, so you receive a credit. I ask you, do you like bacon? Yes or no? That's a survey question. You can answer either yes or no, and you are going to receive credit for it. There are take-home poll questions. Actually, there will be 2 of these take-home poll sets for next week. One is about serial dilution, the other one is about the syllabus. And they will be assigned at the end of the semester, end of the lecture. I'm actually going to post them today, and you will need to complete them. So before you leave, I need to have 2 volunteers, please, with a piece of paper and a pen. The faster you volunteer, the faster you get out of here. That's how it works. Okay, 1 volunteer and another volunteer, please. Yes, just bring a piece of paper and a pen, please. The rest of you, please stay put. We're going to be quick, I promise you. I need 1 more volunteer, please. Okay, sir. By the way, I always go by that clock in the back, so we will start by that and we will finish by that. So, very quick question. Please stay quiet so we can finish this. Thank you very much. At the end of meiosis II, you find 2 daughter cells, 4 daughter cells, 6 daughter cells, 8 daughter cells. Can you guys just pick one answer? Just write it, hopefully the correct answer, and just write it on your piece of paper. Excellent. All right. So what's your name?
Mary.
Mary. And your name? Matt. Matt. So we have Mary and Matt here. All right. Both of them answered the question. And let's assume both of them answered it correctly. All right. Mary decided to log into Poll Everywhere and answer the question. So Mary, can you walk up to me and bring me the question, the answer?
I'm not sure.
Thank you. That's fine. So I received Mary's answer, right? So Mary is going to receive credit for her answer. Matt, on the other hand, answered the question also correctly, but forgot to sign into Poll Everywhere. Can I see Matt's answer? Can I see Matt's answer? Say yes or no. No, it's in his hands, right? He answered it correctly. He's awesome. He knows biology, but I cannot see his answer. So think about this exercise every single time when you log into Poll Everywhere or you forget to log into Poll Everywhere, because I can only see your answer if you log in. Thank you for your attention. Thanks, Matt. Thanks for it.